All right, so get ready for a deep dive today into some seriously cool research happening in breast cancer treatment. We're zeroing in on HER2 positive breast cancer, the aggressive kind. And get this, researchers might be on the verge of a major breakthrough.
Yeah, absolutely. We've made some good progress, but drug resistance, it's a real hurdle in treating HER2 positive breast cancer. That's what makes this research so interesting.
For sure. We're talking about findings straight from Cancer Research Communications. That's from the American Association for Cancer Research, in case you didn't know. They're looking at this new drug, Tucatinib, paired with a treatment we already use, TDM1. And what's got me hooked is this isn't some pie-in-the-sky idea. Tucatinib is already showing serious potential out there in the oncology world. So for anyone listening who knows how important HER2 positive breast cancer is, this is a big deal.
What's really neat is this research, it's not just theory. Tucatinib is already showing it can really work in actual clinical trials,
Okay, so before we go too far, let's break it down. What makes HER2 positive breast cancer so tough to treat?
Basically, it comes down to these things, HER2 proteins. In HER2 positive breast cancer, these proteins are like, well, imagine accelerators stuck on go, making those cancer cells multiply like crazy.
So treatments like TDM1, they try to target these like overactive accelerators. But like a car that finds a way around a stuck accelerator, the cancer can become resistant, am I right?
Perfect analogy. Over time, those cancer cells, they can become resistant to treatments like TDM1, and that's where this Tucatinib comes in.
From what I've read, Tucatinib is what they call a TKI. That's short for Tyrosine Kinase inhibitor. It's like a wrench, messing up the signals within the cancer cell that tell it to grow and multiply. The cool part is it tackles these out-of-control HER2 proteins, but in a totally different way than TDM1 does.
Yeah, exactly. If you think of TDM1 like a smart bomb zeroing in on those HER2 proteins, Tucatinib is more like a sneaky saboteur messing up the signals those proteins need.
So two different game plans, same target. Already sounds promising, right?
Mm-hmm.
But then the synergy they found, that's what really blew me away.
That's where things get really interesting. In some cases, putting these two treatments together doesn't just add their strengths, it's like it multiplies them. This synergy thing means you're getting a bigger effect than either drug could manage on its own.
That's wild. So it's not just about using two treatments, it's about them working together to really boost their impact. How does Tucatinib actually do that, though? What's the secret weapon?
Well, now we're diving into the nitty-gritty of how cells work. Get this. Tucatinib actually makes more HER2 show up on the surface of cancer cells.
Hold on, more HER2. Isn't that like adding fuel to the fire?
You'd think so, right? But here's the catch. Tucatinib increases a particular kind of HER2, the inactive type. Think of it like setting out decoys for TDM1 to lock onto.
Oh, I see. So you're kind of tricking the cancer cells with a fake target, letting TDM1 deliver the knockout punch.
Exactly, and it does this whole thing through a process called ubiquitination. Now, normally this process is like a tagging system. Cells use ubiquitination to mark proteins for destruction. But Tucatinib interferes with that, so those inactive HER2 proteins don't get broken down. More of them stick around for TDM1 to bind to.
Wow, that's some next-level cellular manipulation. Using the cancer's own system against it, brilliant. This all sounds incredibly promising, but how'd this combination actually work outside of the lab? Did they see similar results in, a living organism?
They did. To study that, they used what they call xenograft models. Basically, they put human tumors into mice to see how the treatments worked in something more, lifelike.
You're probably wondering, did this combo pass the test? Drum roll.
In most cases, absolutely, yeah. We're talking about tumors shrinking significantly. And get this, sometimes they even saw complete responses. That means the tumors completely vanish.
Okay, now we're talking. Complete responses, that's the dream, right?
Yeah.
So this combo could really be a game changer for folks with HER2 positive breast cancer.
It definitely looks that way, super promising. But we have to remember, cancer is tricky, complex. In one model they tested, the results weren't as impressive.
Oh, what happened there? The combination, did it just not work at all in that case?
It's not that straightforward. In that particular model, TDM1 by itself and the combo therapy, they both had kind of limited effects, similar to each other. So maybe that model itself, not the treatments, might be resistant to therapies targeting HER2.
So even with promising research like this, cancer can still throw a curveball.
Sadly, that's how it goes sometimes with this disease. Always more to uncover.
I guess that's why they call it research and not a done deal. Seeing this combo work so well in those other models is still super exciting. But before we get too far ahead of ourselves, let's dig into how Tucatinib pulls off this cellular magic trick. We're going deep into the science now, folks.
Absolutely. Understanding the little details of how it all works, that's key to really appreciating what this research could mean.
So we've learned that Tucatinib helps out TDM1 by making more inactive HER2 plop up on those cell surfaces. But the research also looked at what happens inside the cell with this powerful 1-2 punch.
Right. There's a whole other side to this research that's worth looking at. Remember we talked about those HER2 proteins acting like accelerators, pushing those cancer cells to multiply?
Totally. Too many of those HER2 proteins spell trouble, driving the growth of the cancer, right? And we know Tucatinib and TDM1 are designed to, interfere with that whole process.
Right. And the fascinating thing is, they each mess with different parts of that growth signal. Imagine a relay race, right? Tucatinib is like tripping up that first runner, stopping the signal before it even starts. And TDM1, it's down the line, ready to take out the runner, carrying the signal to the finish line.
Okay, love that analogy. So a double whammy approach to shutting those growth signals down. What did the researchers find when they looked at the combined effect on the signaling pathway? Did it work?
So they used this technique, Elisa. It basically measures those proteins involved in the relay race, how much of them are there. They compared the combination therapy to each drug alone and could see how well the treatment was actually disrupting those growth signals.
Okay, and what they see? Growth signals weaker with the combination.
Way weaker. Combining Tucatinib and TDM1 made those key proteins much less active compared to using either drug alone. It's like cutting off communication at different points in that relay race, right? The message to grow never reaches the finish line.
So you're not just stopping the signal at the start, you're making sure it doesn't sneak through some other way.
Exactly. This multi-pronged attack, it seems way more effective at really hitting those cancer cells' ability to grow and spread.
Amazing. But did they just look at this in a petri dish, or did they actually, check what was happening in a more real-life setting?
Oh, they took it a step further. They looked at actual tumor samples from the mice that got the combination therapy.
Whoa, so they went microscopic, looking inside the actual tumors.
Yep. They used something called IHC. It lets them actually visualize those signaling proteins within the tumor tissue, and the results, they were pretty clear.
Let me guess. Same story. Those signaling proteins were way less active in the tumors treated with the combo.
You got it. Those tumor samples from the combo group, they clearly showed those proteins were being put on pause. Strong evidence that the combination was messing with those growth signals fueling the cancer.
That's incredible. But in the end, what we really want to know is, does this actually mean fewer cancer cells?
The million-dollar question, right? And the answer is a big yes. They looked for specific markers in the tumor samples. These markers show how fast the cancer cells are dividing and whether the treatment is pushing those cancer cells to, self-destruct.
So they were looking for proof that this combo was both slowing down the cancer's growth and killing off the cancer cells more effectively.
Exactly. And what they found was super encouraging on both fronts.
Okay, tell me more. What did they actually see?
Well, the mice treated with the combo therapy had a big drop in those markers associated with cell division, meaning those cancer cells weren't multiplying as fast. And get this, they found a huge jump in markers associated with apoptosis.
Hold on, apoptosis. Refresh my memory. What was that again?
Basically, apoptosis is programmed cell death, like a self-destruct button inside a cell. It's totally natural, keeps our bodies healthy by getting rid of old, messed up, or potentially dangerous cells.
Right. So you're saying they saw more of this self-destruct button being pressed in the cancer cells that got the combo treatment.
Exactly. Those markers were going up, telling us that Tucatinib and TDM1 together were basically telling those cancer cells to kick off their own self-destruct sequence.
Now, that's what we like to hear. So putting it all together, this research shows that Tucatinib and TDM1, when used as a tag team, they're a force to be reckoned with. They mess with those crucial HER2 growth signals, both inside and outside the cell. But not only that, they also slow down how fast the cancer grows, and this is key, increased cancer cell death.
It really shows how figuring out the little details of how cancer works can lead to really creative and effective ways to treat it.
For sure. This research is seriously exciting, and it seems like the potential for this combo therapy is massive. Now, I don't want to get ahead of ourselves, but this research, they also dug into the blood-brain barrier. That's a notoriously difficult thing to get past when treating brain metastasis. And what they found there is pretty remarkable. So we've been talking about how effective this Tucatinib and TDM1 combo is against HER2-positive breast cancer. But you brought up this blood-brain barrier thing. For anyone listening who's like, what's that? Well, why is it such a big deal for treating cancer, especially in the brain?
Think of the blood-brain barrier like, a really good bouncer for your brain. It keeps the bad stuff out, which is great. But it can also make it really hard to get some medicines into the brain right where they're needed. That's a huge problem for treating those metastases that spread to the brain because it means a lot of promising treatments just can't get to the cancer cells to do their thing.
Makes sense. So what did the researchers find when they tested this combo's ability to get past that barrier? Could it actually make it through?
This is where it gets super cool. They found that Tucatinib, just by itself, it can actually cross the blood-brain barrier. That's huge because it means it might be able to directly target those cancer cells that have, traveled to the brain, something a lot of other therapies struggle with.
Okay, that's a big deal. Does that mean Tucatinib could be a game changer when it comes to treating these brain metastases in HER2-positive breast cancer?
It definitely has the potential. In their studies, Tucatinib alone, it actually worked better than similar drugs at treating brain metastases in those mouse models. They also wanted to see if adding TDM1 would make it even more powerful,
And what happened? Did the combo really pack that extra punch against those brain metastases?
Well, in this particular study, the combination definitely worked, but adding the TDM1 didn't seem to make the Tucatinib that much stronger against those brain metastases.
Interesting. After seeing how well they team up in other places, kind of surprising that TDM1 didn't really move the needle in the brain. Any ideas why that might be?
It's not totally clear yet. One thought is, even though Tucatinib can slip past the gates, so to speak, TDM1 might still have trouble getting to those brain cancer cells in high enough amounts to really make a difference. Maybe there's other things going on that make it less effective in the brain.
So, still a lot to learn, especially about how these drugs work in the brain. But just the fact that Tucatinib can cross that blood-brain barrier at all, that's a huge win.
Absolutely. It opens up a ton of new research possibilities and potential treatments for people dealing with HER2-positive breast cancer, especially those with brain metastases.
It's really amazing to see the progress being made. This research shows how getting into the nitty-gritty, the little details of cancer biology, can really lead to amazing breakthroughs, the kind that can really change lives.
It really highlights how powerful scientific exploration can be. Every new piece of the puzzle we find, every discovery we make brings us closer to, outsmarting this crazy complex disease.
Totally agree. This has been a deep dive for sure, packed with exciting findings. For everyone listening who wants to really get into the weeds with this research, we'll make sure to link to the full study in our show notes. But before we wrap up, I want to leave you with something to think about. We've seen how taking existing therapies and, trying them together in new ways can give some incredible results. So here's the question. What other awesome combos are out there just waiting to be discovered, and how will they change the future of cancer treatment?
Now, that's a question worth pondering for sure. The possibilities are pretty mind-blowing when you think about it.
They really are. And on that note, we're wrapping up another deep dive. Thanks for joining us on this little adventure into the science. Until next time, stay curious, everyone.